session today. You have an evaluation form. Please go through, look at what you're evaluating before they start evaluating, before they start the presentation. And the grade for the presentation will be 25% weighted of your own score of yourself. So whatever you score is 25% weight. Your score combined of the presentation will be 25%, and I'll have the 50% weight of the grade. Okay, so please be as fair and as objective as you can. So we have a time limitation. It's 20 minutes. So hopefully you adhere to the time. And there will be 5 to 10 minutes questions. If you ask a question, that goes towards your participation point. And if you already asked all possible participation questions, it will be an additional +2 for your asking question. We want to be engaged. You're supposed to read these manuscripts. You're supposed to pay attention to the presentation. It's part of the course requirement. It reinforces what we learn in class. So you are really responsible for the material. So ask questions, and this will help them also show that they understand the material and they can answer the questions to the best of their ability. So that's the first part of today's lecture. We're going to spend about half an hour, 20 minutes presenting and up to 10 minutes questions, and then I'll start with the lecture after that. So before we get started, I just want to tell you, sorry, I kind of messed things up. Instead of posting the legume protein lecture, I think I posted the daily. What is that thing? I posted the daily protein lecture twice, or the milk protein lecture twice, but I corrected that. So I did post the milk protein lecture in color. So if you go back here, you'll find the lecture slides now in color. And then not this week, the week 11 is where you'll find the soy or the legume protein lecture slides. So there are important additional reading material in addition to the chapters that I have at the top of the page for each topic. These additional reading material will be part of the instruction for my lecture piece. And if you go in there, you'll find 3 articles. And I actually underlined what key information that you are responsible of knowing and interpreting the results. I'll talk about most of them in lecture. So as you can see, I underline the important information or the relevant information, and I talk about them in lecture. So this is kind of your additional study material. So 3 articles in total. Okay, so that, and last thing, no. So the last thing is next week you can turn in the practice problem. Okay? And then once everybody turns it in, I will show the key, and then after that there will be another set for the week after next. And I think that's the last practice set. Okay. So, you guys ready? As ready as can be?
As ready as can be.
All right. Oh. All right, so you have this. You can go forward and backwards, and then you can point. And please use this or be close to the computer.
Okay. So, our presentation is on bioactivity of whey protein hydrolysates. And so before I can go into our papers, I want to give a little background. So for bioactivity of whey proteins, so we know that whey proteins consist about 20% of total milk proteins in milk. And so that includes the beta-lactoglobulins, alpha-lactalbumins, BSA, immunoglobulins, and other minor proteins. And they're also rich in amino acids such as leucine, isoleucine, that are part of rich amino acids, essential. And so further along, bioactive peptides are typically short 5 to 11 amino acid sequences. And so those are generated by enzymatic hydrolysis. So here's like a little diagram, it's only 2 or 3, but we have the whole peptide chain, and then through enzymatic hydrolysis, those peptide bonds are cut to create smaller sequences. And so from our papers, we see that the profiles of these peptides that are generated are dependent on what type of enzyme is used, the hydrolysis conditions, and the substrates that are, whether it's already partially hydrolyzed or concentrates, isolates, etc. And so studies have shown that bioactive peptides have benefits towards the immune, cardiovascular, nervous, and for our case today, gastrointestinal systems. So before I continue on to the papers, there's also 2 essays that were done that are common between the 2 of them. So I just want to give a little background. They're not really in our class, but for one of the essays, DPP4, enzyme inhibition. And so that is dipeptidyl peptidase 4, which is an enzyme that cleaves insulin hormones that are found in our gut, gastrointestinal digestion. So this was employed as an in vitro indicator for anti-diabetic effects. And so this assay actually measures the potency of how much protein is needed to inhibit 50%. The other one is oxygen radical absorbance capacity, which is a measure of antioxidant capacity of the peptides, which can have inhibitory effects on radical scavenging. And so this is important because type 2 diabetes, those patients have a higher oxidative stress status. And so for our first paper.
Now I will introduce the first paper, the enzymatic generation of whey protein hydrolysis under pH-controlled and non-pH controlled conditions, impact on physical, chemical, and bioactive properties. It is published in 2015. And first is the justification. The research is focused on the whey protein hydrolyzed by 2 different enzymes under different pH conditions. We know from our class that all of the enzymes used in food application are protein. So the tertiary structure of protein depends on many factors like temperature, salt concentration, pH, etc. to optimize enzyme kinetics. Therefore, the hydrolytic activity of enzymes may be affected by even a small changes of these factors. For example, the pH changes which may generate different peptides during hydrolysis process. The generation of the different peptides may impact the physical, chemical, and the bioactive properties. So the overview of the paper, the purpose of the research compare the impact of whey protein proteolysis under controlled and uncontrolled pH conditions of 2 enzymes and determine the impact on physical, chemical, and bioactive properties. Now I will introduce the experiment specifically. The first is enzymatic hydrolysis of whey protein. The substrate used is the 81% whey protein concentrate, and it was resuspended at 5% in distilled water and rehydrated at 50 degrees C for 30 minutes under agitation, and then adjust the pH for a specific enzyme. The enzyme used are the papain, and another one is a microbial-derived alternative papain-like enzyme. For papain, adjust pH to 6.3, and for papain-like, adjust to the 6.1. After the ratio of the enzyme and the substrate is 2%, After adjust pH and add the enzyme, the hydrolysis process were carried out at 50 degrees C for 3 hours, and then it was terminated by heat inactivating the enzymes in a water bath at 90 degrees C for 20 minutes for papain-like, or 100 degrees C for 40 minutes for the papain enzyme. And then the sample were freeze dried and stored at minus 20 degrees C. And then the sample analysis. The degree of hydrolysis, molecular mass distribution, peptide profile of whey protein hydrolysis, bioactive properties were analyzed. The first is the degree of hydrolysis. We can see the table first. This rate is using the papain as the enzyme to hydrolyze the sample, and with different pH regulation, we can see it's the degree of hydrolysis is very similar, and this rate is using the papain-like enzyme. There's no significant difference between same samples with different pH regulation. However, the whey protein hydrolysis, hydrolyzed by the papain-like, this part, were significantly more hydrolyzed than the samples hydrolyzed by the papain. And then the molecular mass distribution. The peptide mass distribution was observed by gel permission (HPLC). It was covered in our class for the protein characterization. Is separated by different size. The size exclusion. We can see this part is a sample by using the papain-like. This is using the papain. We can see the sample hydrolyzed by the papain-like has a higher proportion of short peptides than the samples hydrolyzed by the papain. And the next part is the peptide profile of whey protein hydrolysis. This graph is our reverse phase ultra performance LC-MS specific chromatograms. The whey protein hydrolyzed with the papain and the papain and the papain-like, and the pH regulate pH to the 6.37 and non-controlled, and 6.17 and non-controlled. We can see in these red dashed boxes, it is indicated some peaks display different intensities under different pH conditions. We can also see this Venn diagram. It shows common peptides. It shows the common and specific peptide peaks. The overlap areas shows the common peptide peaks. We can see most peptides were common within the whey protein hydrolysis samples with the same enzyme preparation, but there were minor differences. However, no clear pattern could be observed concerning the effect of each pH conditions on the intensity of the peptides released. Another part is the bioactive properties. The first is DPP4 inhibition. The IC50 means the concentration with 50% inhibition of DPP4. The less concentration needed means the more potent. So we can see this one and this one. It shows that the whey protein hydrolysis is more potent than unhydrolyzed whey protein. And there's no significant difference between the pH condition of whey protein hydrolysis hydrolyzed by papain, but there's we can see these three. There's a significant differences among conditions for whey protein hydrolysis hydrolyzed by the papain-like enzyme. And another one is for testing the antioxidant properties, the ORAC values. The higher value means higher antioxidant activity. So we can see the whey protein hydrolysis has a higher antioxidant activity than the unhydrolyzed whey protein. And there is a higher values for whey protein hydrolysis at constant pH 7. This one is 285 and 308. And the conclusion for this paper, the whey protein hydrolysis generated with or without pH regulation showed similar physical-chemical properties but had different bioactivity potency. Bioactivity was dependent on the enzyme used for hydrolysis and the pH of the system. And the last part is what we think. First is, although no differences were noted in the physical-chemical characteristics, the study indicates that the conditions for protein hydrolysis had a large impact on the bioactivity of peptides. And this study lacked peptide sequencing. Therefore, any future studies should identify the specific peptides and their connection with the bioactivity. And samples were freeze-dried. Is very expensive, so for the industrial application. And the papain-like enzymes had a higher degree of hydrolysis and smaller peptides. In food application, it may be result in highly perceived bitterness. So this interests medicine.
Yeah. And so for our second paper that we looked into, it was in vitro bioactive properties of intact and enzymatically hydrolyzed whey protein and targeting the intro-insular axis. So, quick overview of what intro-insular axis and why it's important was that, and so they're focusing on type 2 diabetes mellitus, which is a chronic metabolic disorder that's currently increasing prevalence with an estimated of 550 million cases by 2030. And so there's emerging evidence, again, that whey protein that's been hydrolyzed have a role in managing type 2 diabetes mellitus. So the intro-insular axis, by definitions, are the hormones and neurotransmitters from the gastrointestinal tract that regulate the secretions of hormones by the pancreas. And so one of those hormones is incretin, which are secreted by endocrine cells, which is in the gastrointestinal tract. And these stimulate insulin activity, which is important for people with diabetes. And so a major hormone example is glucagon-like peptide 1. And so with the DP4 enzyme, they cleave the GLP-1 enzyme, which is important. So measuring the potency of inhibition is part of that. And also, type 2 diabetes mellitus also compromises the body natural antioxidant defense. Therefore, it's important to see how these hydrolysates and bioactive peptides are with combating antioxidants or radical scavengers. So the purpose of this was that they evaluated commercially available intact and hydrolyzed whey protein and seeing how those proteins interact with the intro-insular axis, so the DPP4 or secretion of the GLP-1s and insulin. So then use the whey protein concentrate, a whey protein that's been hydrolyzed 32%, and a whey protein has a degree of hydrolysis of 45. So sample analyses. To test an in vitro simulation, they did a simulated gastrointestinal digestion. Physical chemical properties, like the other paper, they did a reverse phase ultra performance liquid chromatography, which is just a higher pressure HPLC, and a gel permeation, which is size exclusion. For bioactivity, they did a DPP4 inhibition, the ORAC assay for antioxidant capacity, and then they did in vitro of insulin secretion by pancreatic beta cells, and then they looked at in vitro GLP-1 and secretion in STC1 cells. So they used cell cultures to simulate what could potentially happen. So for the simulated gastrointestinal digestion, they used freeze-dried samples of the intact whey protein concentrate and the hydrolyzed whey protein. They diluted that to a 2% whey protein, and resuspended it for 30 minutes at pH 2, which is for the pepsin digestion, at a 1 to 40 enzyme to substrate, and they did that for 90 minutes. After 90 minutes, they took a subsample out, which I believe is for a blank, but yeah, and then they further did the digestion with correlase PP at 150 at 7.5, and then heat inactivated that. And they proceeded to cool the digested hydrolyzed protein to room temperature, froze it at -20, freeze-dried, and then continued to store it at -20 until further use. So for first physical-chemical properties, so the result shows the differences between whey protein concentrates and the hydrolysis afterwards. So we can see from whey protein concentrates, it contains most of the major whey proteins. And from 32% and 45%, you see that greatly decreases from hydrolysis. And then after the simulated gastrointestinal digestion, we see that most of the major bigger peaks, at least for the whey protein concentrate, has degraded to smaller peptides, and same as the 32 and 45, and they are towards the more hydrophilic regions, indicating that these peptides, there's probably more hydrophilic residues being let out. For molecular mass distribution, size exclusion, we see that it's pretty much what we would expect. So whey protein concentrates had a higher mass distribution above 5 kilodaltons because it hasn't been hydrolyzed yet. But here, after hydrolysis, we see an increase in the smaller mass distribution because the peptides are being broken into smaller pieces. And then after simulated gastrointestinal digestion, there's a large increase in the smaller ones, such as for 45, we have a 96% of less than 0.5 kilodaltons. So for the DPP4 inhibition, we see that the hydrolysates have a lower DPP4 value, IC50 value, saying that it's more potent than compared to the whey protein concentrates. And then comparing the whey protein concentrates to the 35, we see that from before and after simulated gastrointestinal digestion, there was a decrease. However, for 45 degrees hydrolysis, before and after, there was actually an increase or a decrease in potency. So from the other bioactivity assays, we can see that this is the trend. For antioxidant activity, we see here that for whey protein concentrates before and after the simulated digestion, there was a 2.5-fold increase, which is great. However, for the 32%, there was not really a huge change, or there's not a significant change. And again, like the DP4 inhibition after simulated gastrointestinal digestion, the antioxidant capacity actually decreased. For the first in vitro simulated hormone, GLP-1 secretion, they use secretin tumor cells, which are also found in the gastrointestinal digestion system. And so they are the ones that secrete the GLP-1. So the results for that shows that for whey protein concentrates, after simulated digestion, there was a decrease secretion. And here we see that hydrolysates, the 32 shows that there's an increase, although it's not significant, but the trend kind of goes for the other previous slides where 45 after simulated digestion decrease. And so this, they consider that could suggest that we would need to protect these enzymes when we eat them from further degradation.
Enzymes?
Enzymes. proteins. Proteins from hydrolyzing peptides. Peptides from further degradation, or too much degradation. And the other one, insulin secretion by pancreatic beta cells. Here we see that compared to the control here, the whey protein concentrates before and after, and the 45 before and after had similar results. But positively, the 32% degree hydrolysis before and after had a significantly larger secretion compared to the concentrate in the 45. But overall, within each sample, after simulated digestion, there was no significant change. So this says that we can see for the GLP secretion that the intact whey proteins without hydrolysis can still secrete GLP-1 incretin hormones. We see that whey protein hydrolysates, again, like the previous paper, can inhibit DPP-4 enzymes and enhance antioxidant activity. And since they did an in vitro version of the simulated gastrointestinal and the results from that, suggests that potentially what they found in this in vitro version of what could happen in our gastrointestinal could be applied in vivo within living beings. And whey protein hydrolysates in general can be suitable as a multifunctional nutritional therapy for management of type 2 diabetes. So samples were all analyzed by freeze drying, as we know is not feasible for industry. But in the paper there was mention of spray drying once, but I'm not really sure where that's used in the analysis. Again, there was no sequencing of peptides before and after the simulated digestion, and so it would help with identifying the potential amino acids that are actually involved in enhancing these benefits, bioactivity benefits, and their mechanism. And so future studies should focus on understanding the interactions between those bioactive peptides and within the insular axis. And here we see that too much hydrolysis can actually yield lower bioactivity. We saw that between the 45 and the 32. So final conclusions between our two papers is that we see that bioactive peptides generated depends on the conditions, such as the enzymes, so from the first paper, pH conditions, or substrates from the 32 compared to the 45, or concentrates themselves. Whey protein peptides have bioactivity that are beneficial towards the gastrointestinal system, so the inhibition of enzymes, capacity, and secretion of hormones. Both our tests were done in vitro, so to confirm that our results will need to be done in vivo. And so with that, the exact mechanisms of how these bioactive peptides work are still unclear, so sequencing would greatly elucidate some of our questions regarding that. And then on a feasible standpoint, smaller peptides in use, even though they have all these great benefits, would customers like it if it's because it would be perceived as very bitter? And then again, freeze drying is not really feasible. I would say spray drying will be a better alternative that's potentially better. So yeah.
Yeah, thank you.
It's over.
Not yet.
Not yet. Questions? Go.
Questions. I have lots of questions, but I will wait for others.
I just had a quick question about Zhao Cheng's paper. Do you know like why they picked papain, or why they would want a microbially produced version, or did they speak to that at all? It's okay if they did.
Not really sure.
So why the choice papain and papain-like?
Yeah.
Did they justify that?
I think just compare the different.
Did they justify the choice of their enzyme?
I'm not 100% sure. Because well, both of them are just proteases, papain-like.
So why did they call it papain-like?
Because it was microbially derived. Yeah.
Organisms. So it's just micro-derived alternative. So maybe some properties is very
So they
similar, maybe.
induce its production in microbes.
Yes.
Mhm.
Okay.
Going off of that, why do you think that they did different temperatures and different times for the papain like?
I'm assuming it's because these enzymes, well, as we know, there's the pH conditions where enzyme kinetics are optimal. So probably with the enzyme, papain-like, I believe, had a higher temperature that's probably more suitable for that one. I mean,
So optimal conditions is different, so
Mhm.
it's also different temperature.
Probably different, since we saw that they had different
Maybe they have a pretest or something.
different degrees of hydrolysis or peptides made is probably because of their structure is different from each other.
I have a question along these lines.
Mhm.
So, for one enzyme, it seems like the pH effect was not significant for the papain.
Mhm.
But then for the papain, like, there was a significance, and also controlling the pH was important. So, what do you think, why is that? Why do you think, how can you speculate this?
I think for the previous one, the physical chemical properties is very similar, but the bioactive properties has some difference. So I think maybe the peptides use the different enzymes. Maybe some specific peptides have some functions. So I'm not sure.
No, my question is related to the enzyme itself.
Mhm.
Why would an enzyme be affected more by pH change than another enzyme?
It could be what I would think is the
It's more sensitive or
sensitive, maybe the exterior amino acids of composition of the, like, comparing for the papain, like, might be more prone to being ionized differently.
Yeah, so you're getting close, you're getting hotter and hotter. So, we talked a little bit about how the active sites
Mhm.
could be impacted by pH.
Yes.
And different active sites, if the papain-like is slightly different than the papain, it could be ionized differently and impacted by the pH differently due to the composition in the active site of the enzymes.
And so, kind of clarifying, they use a pH stat to control the pH, so if it were to, like, dip down,
Yeah.
it would adjust to come back up. And then there was a non-controlled version where they would just let the hydrolysis occur without any control. So over time, the pH could change, and they wouldn't change it. They wouldn't bring it back to what it was originally at. So they were seeing how that could happen. So, like, on an industry standpoint, by controlling pH, you're probably introducing a lot of salts and stuff like that. But where I believe, like, they normally don't really control pH when they do protein hydrolysis, right?
In industry?
In industry?
Well, I don't know. I have no idea
Yeah.
if they actually monitor pH during
Yeah.
hydrolysis, or they have it in a chamber
yeah.
and
It just happens.
sometimes it's continuous too.
Mhm.
So sometimes the enzyme is immobilized and the system just flows by.
Yeah.
I
So
don't
in that case, it wouldn't be controlled. So we were seeing there was, like, a difference in bioactivity between the two type of conditions.
Other questions?
Do you think it was sufficient to measure ORAC as an overall antioxidant capacity as compared to its actual antioxidant activity in vivo?
In vivo. I guess, okay, so with that was the fluorescence method. ORAC. It's comparing, like, how they did it outside and within.
Yeah, so you're looking at ORAC's like overall capacity as
Mhm.
compared to like actual antioxidant activity.
I guess I didn't really look into, like, how antioxidants could be measured differently from capacity.
Because different, building on
Mhm.
that question, different assay determine activity by a different mechanism.
Yeah.
So it is radical scavenging
Mhm.
or metal clading or what's the mechanism
Mhm.
of each assay, and how does this compare to
Yeah.
what
So I
actually
would
happens?
say because they didn't do like any sequencing, they really didn't know exactly what peptides were being made, and so maybe they were just looking at the overall arcing antioxidant capacity, and maybe furthermore.
But just doing ORAC is not enough
Yeah.
to look at all different mechanisms.
Yeah. And so for at least the second paper, that was the first time they did those four all together. And so they were looking at different parts. And so they could have added more, but that paper alone was a lot. Especially with what they were doing with cell cultures for the secretion of those hormones.
So in the DPP4 inhibition, they
Mhm.
actually got the high, if I'm understanding right, the
Mhm.
higher the number, the more it's inhibited?
No, the higher the number, the more protein it takes to.
Oh, to inhibit?
Yeah, so the more is meaning it's less potent.
Less potent.
Okay, so you want a lower number?
Yes.
Okay.
That took, when I first looked at that, I was like, what? And
That's the
then
IC50.
that's the IC50.
So the amount of protein that will cause 50% inhibition.
Inhibition,
So
yes.
if less protein causes 50% inhibition, that's more potent.
Yes.
Gotcha. Okay.
So we saw that in like at least the 32 or the hydrolyzed, those were lower.
So even though you have, after digestion, it
Mhm.
goes up, I mean, it's really close.
It's very close.
There's not, I mean.
Yeah, for at least the 45. That was just kind of the trend that we saw in like the other ones, that after simulated digestions for the 45, there was like a decrease in beneficial bioactivity. So that to me kind of suggests that if you get too much hydrolysis, it might not be beneficial anymore.
Okay, I'm gonna ask. So you mentioned at first, your first background in
Mhm.
generation of, or the hydrolysis itself. Do you think it will be different if the product is hydrolyzed in vitro versus if we consume whey protein as such,
Mhm.
and it's gonna be hydrolyzed in our body?
Mhm. So that's
how is it different? Yeah.
how is it different? I would say for like when we do it in vitro, we have one specific enzyme, or maybe a couple doing the hydrolysis, where in vivo within our bodies we probably have a lot more different
So
proteases.
if you wanna differentiate it in one word,
In
what
one
would you
word.
use?
Complex.
Controlled versus non-controlled
Ah.
hydrolysis?
Yeah.
So outside the body, you generate these protein hydrolysis with
Mhm.
a targeted outcome.
Yes.
So you would use specific enzymes, specific conditions that will give you the
Mhm.
most potent
Yeah.
activity, and you hope it won't be degraded more during
Yes.
digestion, which your second paper showed that actually in some cases it did degrade.
Mhm.
What happened? Why do you think activity was lost?
Activity was lost. So
After another digestion.
after another digestion. Oh, so from the so the commercially available ones, the 32 and 45, and then after the simulated gastrointestinal. So I could, well, what I would think is since they're commercially obtained, they all, well, there's actually a table in the paper that I took away to save some time, where it showed the total amino acids within each of the different commercially available, and you can see there's a variability between the three. So that could be a cause of why there's different bioactivity, because they have different peptide amino acids in each of them, which kind of made me confused why the amino acid composition of a whey protein would change over time.
Depends on the source.
Yeah. So the sources were all different. So that's one
But
critique.
after second digestion,
Yeah.
amino acid composition should not.
It should not change.
You're starting the
Yeah.
same material.
Mhm. So.
What I think I was hinting towards is that, say you have a potent peptide,
Mhm.
and let's say it is susceptible to further digestion, and you hydrolyzed it more, so you
Mhm.
lost its potency.
Yes.
So you have like 5 amino acids in a peptide, and it was digested even more,
To agree.
broken down to free amino acids,
Mhm.
or broken down to
Thus
dipeptides,
losing
then
its-
it would lose its potency.
Mhm. Yeah.
Yeah. Okay. Did they talk at all about the origin of the whey protein concentrate that they've used in terms of its molecular structure? Is it denatured? Is it Did they talk about anything, or did they just say
They,
commercial whey protein concentrate?
for mine, it was commercially obtained.
And
So.
for mine is
Did they produce it, or did they?
just not have the structure.
They did not
Still
provide.
produce.
Okay. I'm gonna call you on what you said about reverse phase.
Oh.
Reverse phase.
Reverse
PLC.
phase. Yeah.
So why do you think that the peptides were more hydrophilic?
Well, let me see if I can go back to it. Oh. So many slides. Okay. So looking here, so after simulated digestion, we have peaks up here, and so reverse phase, the mobile phase is polar. So we see that after simulated digestion, these peaks are more intense earlier in the chromatogram.
That could be just because they're smaller peptides and
Oh.
they have more relative to their size, carboxyl and.
Okay. So my thought was potentially it could be because there's more hydrophilic amino acids being exposed and they're coming out faster.
Could be, but
Mhm.
you cannot make that general statement. It's
Okay.
Mm-hmm.
because they're peptides, so they have free amine and free carboxyl groups
Also,
relative to the large.
so they're more polar, right?
More-
Because those free
Relatively, I would
relatively
say.
more polar.
But if you want to compare among the peptides
Mhm.
themselves, we cannot really tell because it's not size exclusion.
Yes.
Coupled
Okay.
with reverse phase, you cannot really tell, is it the size they're too small, or it could be the one peak is not as small as the week that comes at 8 minutes, the one that comes at 5, let's say, is not as small, might be larger, but released more hydrophilic.
Yes. Okay.
So you have to be very careful on what you conclude out of the retention time for reverse phase.
Okay. Yeah, I was just using the general concepts that I knew.
Yeah.
Okay.
Okay, I have more questions, but I'm gonna cut it short because time.
You can ask us later.
Yeah.
Ask us one-on-one.
Alright.
Okay.
Thank you. Courageous group to go first. Yeah, okay. So before you leave, just give me your evaluations, please. Alright, what I'll do is I will try to finish the dairy protein and take a break and then start the soy or the legumes. Smiling. No eye contact. What? Nothing? Okay. I just wanted to review with you these differences. So, what's the difference in amino acid distribution between casein and whey protein?
Casein, is it that? So casein amino acids are randomly coiled, open structure kind of? Oh, that's much of a structure.
Mm.
Okay.
Highly amphiphilic, so they have clusters of hydrophobic and clusters of hydrophilic amino acids, whereas in whey it's more random and regular distribution. So molecular structure, it's casein open, random, barely any secondary and tertiary structure. It's flexible, whereas whey protein is globular, highly structured protein. What about stability and gelation?
Casein is very stable.
Very stable, and whey protein, in order to make a gel out of it, you have, yeah. So stable up to really high temperature, 140 degrees C for 20 minutes. And you do get age gelation over time due to the beta-lactic globulin being denatured, interact with kappa casein, and then you have also hydrolysis of beta-casein by endogenous enzymes, so they coagulate and precipitate. And then the enzyme gelation is the curd formation, like cheese making. So association, we have casein, they form this unique micelle, whereas whey, they're mostly dispersion. They could be monomer, dimer, or octamer based on the pH of the system. What about enzyme sensitivity?
Sensitive to chymosin.
Yes, so yeah, casein is sensitive to chymosin. It's more susceptible to enzyme hydrolysis in general, given also its open structure, whereas whey proteins resist hydrolysis since it's compact and globular form. That's why babies, in order to have the infant formula substituted with whey protein, usually it's whey protein hydrolysis, because they don't have very well developed digestive system to digest whey protein as such. Surface adsorption and emulsification comparison.
It's high.
Yep. So we have flexible open structure, highly amphiphilic like we said here, and high surface adsorption, whereas the native form of whey protein is low surface adsorption given to the low surface hydrophobicity of a native whey protein. What about solubility?
We see in precipitate at 4.6.
Okay. Precipitate at 4.6, whereas whey protein is soluble over a wide range of pH unless it is denatured. Conjugation. What do we have? The casein proteins, are they conjugated? Phosphorylated, glycosylated, whereas whey protein, no. Do we have sulfur residue? What about that?
Whey is higher than
Whey is higher than casein. So we have very little sulfur containing amino acids, and whereas whey protein is rich in sulfur. Okay.
I have a quick question about terminology. Is amphiphilic, is that the same thing?
Amphiphilic, yeah. They're interchangeable. Yeah. Okay. So again, just a reminder that you can produce different protein ingredients starting from skim milk using different processes. If you use yeast and calcium and change the pH, you get a co-precipitate of both casein and whey proteins. They call it co-precipitate. If you subject to rennet enzyme to produce cheese, you get the sweet whey, and the whey can be concentrated either by heat, you get the lactoalbumin, or you can do ultrafiltration, diafiltration, you can get concentrate and isolate depending on what you put in sequence, and then the ion exchange, usually you get an isolate with that. If you adjust the pH and you get the lactic acid casein, then you can neutralize using either calcium hydroxide or sodium hydroxide, and you get sodium caseinate or calcium caseinate. So in terms of milk uses, so if you have the acid casein, the differentiating acid casein versus rennet casein, the acid casein obviously is insoluble at pH 4.7 or 4.6. It's at the isoelectric point. It's low in calcium and phosphate. Remember that when we said when the pH decreases, calcium and phosphate leaches out of the casein micelle. So then you end up with the low calcium and phosphate casein coagulate. You can resolubilize it by changing the pH. You can neutralize the pH, and it will become soluble again. The rennet casein, on the other hand, is insoluble at neutral pH because the kappa casein has been hydrolyzed. That hairy structure, the glycomacropeptide, is being cleaved, so you don't have any more stability due to the net negative charge of the glycomacropeptide. Therefore, it's insoluble at pH 7.1. It's actually high in calcium and phosphate because proteins you have the bridges, calcium phosphate bridges, that stabilize the curd structure. And if you want to solubilize it, you would want to remove calcium. So you sequester calcium out, then it will be solubilized. Both are heat stable, good flavor, and a high nutritive value, protein efficiency ratio value of 2.5. So there are several applications for these kind of caseins: breakfast cereal, snack and pasta products, and baked goods. So it could be for flavor, structure, and nutritive value additions. So looking at the caseinates, specifically, they're very, very different. So the calcium caseinates, they are cross-linked with the divalent calcium, where the sodium caseinates are not cross-linked because sodium is not a divalent ion. They form true solutions in water. They're soluble, whereas the calcium caseinate, they are actually colloidal substances. So you have suspensions rather than a true soluble solution. The functionality is different. That this has, for example, opaque color versus translucent, low viscosity, high viscosity, very good emulsification, and it disperses well. So if we look at application due to the different functional properties, so whipped toppings, creamers, imitation cheese, whereas the sodium caseinate, infant formula, nutritional beverage, meat products, even baked goods like muffins, for example, you can use sodium caseinates.
Imitation cheese.
Imitation cheese, Alex. What is imitation cheese? So it's not made by the general process of curd formation.
Really like those American cheese, like the processed ones?
That'd be processed cheese. So I don't know if there's something that's worse than processed cheese.
Oh, is it vegan cheese?
Maybe.
Yeah, and they add,
Vegetable protein.
but then it no longer becomes
That's
vegan
not
if you
vegan.
add.
Yeah.
Yeah, it's just, Tonya would know better.
Yeah.
Yeah.
Interesting.
Okay, so here is an article that looked at the functionality of different forms of casein ingredients. In this case, what they did is they processed skim. I took this from the article, it's their fault. I did not create this. Okay. So they take the skim milk and they process it in two different ways. They either produce micelles that are intact micelles through microfiltration, where they remove the whey. They call it natural whey because it wasn't subjected to any pH or heat treatment or enzyme even. And they retain the casein micelle with minimal structural impact on the structure. They do freeze drying and they get the dehydrated casein micelles. And then the next process is they go through the regular coagulation with the rennet enzyme and they produce the curd, the casein curd. They resuspend in water or alkaline solution at pH 7 and they dry it and they call it casein cloth. And they compare both to a commercial casein. They don't say much what the commercial casein source is or how it's produced, but they compare the three together. In this case, they looked at foam expansion and stability, and there is some significant difference if you look at the micellar casein versus the commercial casein. So it seems like the micellar casein behaves better at pH 4, but its functionality reduced a little bit at the different pHs. and they looked at the impact of salt. So if you add salt, how would that impact the functionality? So for the commercial casein, based on how it's produced, it seems to be the least functional at pH 4 versus 6 and 8. So they're both very different. In terms of stability, the opposite. So at pH 4, the micellar casein is closer to its isoelectric point. So if we want to look at what happens to the structure, what happens to the charge, and the stability of the foam. So if you have a lot of no charge, the proteins surrounding two air bubbles are going to come closer to each other and coalesce. But when you have charges, then you will have better stability. So although they form better foam because they're more hydrophobic on the surface, they are not as stable because that hydrophobicity on the surface and less charge will cause them to coalesce at that pH. Whereas the commercial, you can see it has, the salt concentration has an impact. So if you add salt, then you are really shielding charges, then you will reduce the stability again of the foam. With no salt, then the charges enhances the stability of the foam. It seems like the micellar casein was not impacted by salt. So this just to illustrate that the way the casein is produced impacts its structure on the surface, and with that it will have different functionality under different conditions. Although it's the same protein, but it was processed differently. They also looked at emulsion capacity and stability. Again, there are they compared commercial to the casein clot that they formed to the casein micelles, or the micellar casein in this case. And they saw that the commercial casein in all cases behaved better in emulsification capacity. They don't specify much why or what the source of the casein, commercial casein was. So I tried to link the structure to the function here, but unfortunately the paper did not state what that commercial casein was. So, but I just wanted to show you this to emphasize that different ways of producing the same protein will impact functionality differently. Okay, so whey protein. So you have the whey proteins that are functional versus the whey proteins that have been precipitated by heat. They don't have really functional properties. They're aggregates, they're sandy and gritty, and they are only used as nutritive enhancement. So they don't have really functional contribution. Whereas those that are produced by membrane filtration or ion exchange, they're highly soluble over a broad range of pH, mostly used in acidic pHs. They form good gels using thermally induced gelling mechanisms. Also in gels, help the gel structure in yogurt, processed meat, baked goods, induced browning, because they do have lactose, especially the membrane. So the lactose and the protein in baked product will enhance the desirable browning color. So here's an article that compared sodium caseinate, milk protein isolate, which is a combination of casein and whey protein isolate in meat emulsion formulation, or meat patty, basically. So the way they formulated that, they used beef and canola oil as a source of fat. And then they adjusted the protein level either 13, 14, or 15%. Accordingly, they adjusted the beef meat amount. And they kept more or less constant amount of the added protein in the 3 different treatments. So what they noted is they looked at fat and water loss And you can see without milk proteins added, you have the highest loss in fat and water, especially as you increase the protein, the meat protein concentration. Whereas if you look at the whey protein isolate, it has the least amount of loss compared, for example, to sodium caseinate and milk protein isolate when you have the highest amount of meat protein. So why do you think is that? Can you think of a reason why the whey protein in this case behaved better than casein protein? When you heat or cook the,
You're denaturing the whey protein so you get
And
the
the-
heat whey.
then you get the gel. So whey protein is better in forming gel. So when they form better gel, they actually entrap water and also entrap the fat as well. So you end up, whereas casein does not really form a gel. Although it emulsifies, and that's why you have less loss in fat, but really compared to the whey protein, you actually, the gel effect, the gelling effect in this case helped in reducing the loss. Okay. Any questions before changing gears? Well, we're still talking proteins, but changing to different.
Did you remove like 4 slides or?
Did I remove 4 slides?
Yeah.
or what they were-
Oh. I don't know why I removed them. I didn't want to remove them. Well, moving on. Did I put a reference?
Yeah.
It would be good reading material then. Oh, I think that was a nice paper. What? I don't know what I did. Maybe I closed without saving at one point. Or I added it to the student copy and did not added it to the presentation copy.
How would that, isn't the student copy made from the presentation?
Well, the student copy is sometimes different because I don't have the animation built into the student copy. So it depends on what I was working. I probably was working on the student copy instead of my copy. Anyway, we need to move on. So take 5 minutes.
Wait for concentration process.
Oh, there they are. It's in the student copy. Was a good paper. Okay, never mind.
Hearing us through thesis experts are so much better because it's hard.
Makes sense.
Oh, is that cat theory? Or that's these thesis? True style.
Sure, there's so much more healthy than
For next year, I put it in for next year. So, okay. Sure, yeah, I won't look at it before Friday anyway.
Okay. So
Friday
if we send
morning.
it to you Thursday night, that's okay too?
Yes.
Okay.
So you guys next week?
Yeah.
Yeah.
I'm getting really stressed out about college bowl.
What's that?
I'm getting really stressed out about college bowl. Come on. So, my original plan was to do solubility tomorrow. But I
Yeah, it was
think I'm
funny,
just
like
gonna
thinking about
Study.
the digestion, because I'm like,
Research.
it's
It's not
so
important as
short.
college bowl.
Because
I
the
was
digestion
just sitting there and I was like having
like,
panic
you start at
Panic
like
daydream.
Panic mode?
8.
No.
Not done.
Now college bowl
until
time.
4 or 5.
These
I
were
think it's like all day. So these are just like really short, like, yeah, junk food. Yeah.
I'm just in lab here tomorrow. We ran out of axle octane in our lab, but apparently Tony's lab has old oxaloxane.
We have what?
You guys have old oxaloxane,
Oh, sure.
so we don't have, I bought them already. We're not
We
using it.
have
Why is it like this?
Oh, what are we talking
Oh,
about?
something else. Oh, what I'm doing right now. So
Okay.
that's the plan. If the isooctane's not in by tomorrow, we'll just use some. I'm not sure how.
Did you ask if you could?
Yeah, I'll ask them. They haven't used it in a while. Well, Amy already used it. Some of it. And whatever I use, I'll. That's why
Yeah.
I need to look into that.
Yeah.
Isooctane stability. If it's not stable, then I
feel like
won't
I
be
was
doing
like
it.
laying in bed yesterday and like had like big
But-
swirling in my brain.
We got more mobile phase solvents. That's good.
Oh, yeah. Yeah.
Isooctane.
You're spending away.
I know. It's like.
I told George, I'm gonna be watching you, George. I can give you my account
I
number.
looked it up.
Organic compounds.
And then I looked up the lobster change, like the color change in lobster. Just double checking.
I was also yesterday before I went to bed. I was looking at green things. I feel pretty good about reading.
Our guard column comes in tomorrow. I'm so excited.
Okay, there's also this four corner. I was looking at like the brewing process yesterday too. Gosh, so many things to look at, so little time. When's the competition? Saturday. Yeah, Missouri's leaving at 5 in the morning on Friday to drive.
Who's leaving?
Missouri is like leaving
Oh.
at 5 in the morning on Friday to drive here, so maybe they'll be really tired.
I know, I was gonna say, that's school to worry about.
We should just keep them out at Fulton or like, I don't know.
We have to delegate somebody else. Who, Mike, and you get to keep them out all night.
Well, then Jen has them.
Oh, that's right.
Maybe you'll come on Saturday.
Let's see.
College bowl
If you guys are out by 10, then I can come in.
I'm
And
Yeah,
not-
see, but you'll put the others keep them out all night.
Yeah.
And we'll like sneak out and go to bed.
It's my mom's birthday on Saturday, that's my out. It's my very valid reason.
Oh, this is musical. I can't wait till it's over.
Stress is very in this room. Okay, let's move on to another protein source, which is an exciting protein source. So I work on it a lot. And we're still working on it. Never ends. Anyway, legume and oilseed proteins. What do you guys know about these proteins?
Insoluble, sorry.
What? They're insoluble? They can be soluble to a certain extent. But can you guess? You got nothing.
I don't know about C, but A and B are true. I don't know if C is true.
It's not true.
There we go.
Yeah.
Is it the other way around?
No, it's actually both can be allergenic. Yeah. It's just, I put it there for fun. So you can say A and B. Yeah, we'll talk more about A and D. Have you heard of this differentiation when you hear 11s, 7s, 15s, 2s. What does that mean? What does S stand for? It's on the slide, Myson. Sedimentation. Okay, so what can you guess out of that?
Plankton has a higher sedimentation coefficient.
Yeah.
Oh.
Okay, this is one. Is there
That's
anything else correct?
A.
Okay, if glycine has higher, then beta-glycine has lower, right?
Yeah.
So all of them.
Oh.
Yeah.
So here's the definition. So sedimentation coefficient is the ratio of a particle sedimentation velocity, means how fast the particle, based on its size, it sediments once you accelerate using, centrifugal force, the acceleration of the centrifuge. That would cause the sedimentation. It's expressed usually in units of time. So if you have higher sedimentation coefficient, that means it's probably, not probably, it is a bigger molecule. It takes longer, or it's slower to sediment at the same centrifugal, or at the same acceleration. So at the same centrifugal force or acceleration, this will take, the legumin will take longer time to settle or sediment than sediments, because it's larger in molecular size. All right. So legumes and oilseed proteins, we have the non-legume oilseed, the non-oilseed legumes, and the oilseed legumes. So under non-legume oilseed, we have the cottonseed, rapeseed, which is canola, the genetically modified version.
Not all canola is genetically modified.
Yeah, so the rapeseed that is genetically modified is canola.
No, you can actually have something called canola that isn't genetically modified.
Oh, is that right?
It's
I
like
didn't know that.
1% of the crops grown in Canada.
Oh,
I just found out.
good to know.
It is feasible. It's unlikely, but possible.
All right. So, and then the sunflower and coconut oil. So the non-oilseed legumes is your favorite beans. So the common beans, the peas, the lima beans, lentils, chickpeas. So these are legumes, but they have a very low percentage of fat. So they're non-oilseed. Whereas the giant oilseed legume is your soybean. And then you also, the peanuts are considered oilseed legumes, and they are high in proteins. And then there are these cute little crafts. They are known as weeds, actually. Did you know that?
It's a noxious weed.
Yeah.
Wait,
Yeah, it's difficult to transport in and out of the country because there's regulations against them.
Because they kind of spread. Yeah, so we're currently working on camelina, thank you Claire, and then we started working a little bit on silphium. We will, if we get that one grant that we submitted, we might be working on pancreas as well. Okay.
Those are fun names.
Huh?
They're fun names.
Yeah, they are. They're fun weeds. But they are really high and relatively high in oil and protein.
So are they grown as crops? I know camelina is, but the other two, the silphium and pennycress.
Yeah, they are. They can be grown here in Minnesota.
For what? For the oil production or for
Well,
like
they are
the-
developing them for oil production,
Oh, okay.
and I'm telling them we should develop them for protein as well.
I see. Okay.
Yeah. All right. So this chart doesn't include these wonderful crops because they're not really very well known right now, other than being weed, obnoxious weed, you call them?
Noxious.
Nauseous?
Noxious.
Noxious.
It's like obnoxious, but without the
Oh, okay. That's well nauseous.
It makes you sick.
So you have your beans, the non-oil seed legumes, basically. They're all very similar in composition. So if you look at dry beans and let's say peas, you see that they're really high in carbohydrates. And moisture, forget about it, it's not important, depends on the harvest time and drying. And then you have about 20 to 24% protein in that range. If you look at the oilseed legumes, you really have high oil and high protein. And this part here, which is carbohydrate, for example, in soybean, what are the carbohydrates in soybeans? Let's see if you know this. Carbohydrate composition in soybeans. Do we have starch, dietary fiber, sugars? What do we have?
Are we talking hulled or unhulled?
Just the bean with hull, with everything. Or dehulled, it doesn't matter. The hull is the outside fiber. So is there starch in soybeans? No, there's no starch in soybeans. So the carbohydrate components in soybeans are mainly your dietary fiber, and then you have raffinose, stachyose, and then very little amount of glucose. If you look here at the nut, ground nut for instance, doesn't have any carbohydrate, a lot of fat. Well, actually, if you take cashew nuts, for example, they have 60% fat. Did you know that the cashew nut, you can actually light it?
Oh, yes.
Light it.
Cashews
Yeah.
last night.
You just gotta light it.
Put a lighter and light it, it's like, oh, a candle. Yeah.
The candle. All I have is cashews.
You'll be fine.
Okay. So, of course, we're not going to focus on oils or carbohydrates. Who cares about those, right? We're just going to focus on the protein here. All right, so in general, if we want to look at plant protein compared to animal protein, you have two main classification of your plant proteins. You have the albumins and then you have your globulins. So the albumins usually are very heterogeneous mixture of proteins. They are really soluble. They do not have a functionality like texture related functionality to the food application. A big portion of them are enzymes, or they can be called structural protein that gives a structure to the seed itself, rather than functionality for what we care for in food applications. So globulins are the functional proteins, and they are known as the storage proteins. I mean, the plant uses, or the seed uses this protein as a source of nutrition for the germ, right? So they store the protein, and that's why it's called storage protein, because they use the seed uses it for growing and germination and all of that. So the globulin protein represent, on ballpark estimate, about 80% of the total protein in the seed, whereas albumin represent 20%. So general characterization of legume proteins, they have, under globulin, you have two types of legume proteins. You have the legumin and the viscellin. If we're just looking at soy, for instance, the legumin is your 11S glycinin, and the viscellin is 7S beta-glycinin. But this is typical of soybean, but it's different in pea protein, it's different in other legumes. But you have the general class of it, legumin and viscellin. So they are responsible for the functional properties in food applications. So general characteristics of the protein fractions. So you have protein fraction based on their sedimentation coefficient. So you have, it ranges from 2, 7, 11, and 15. So the 2s are your low molecular weight, such as trypsin inhibitor, or a fragment resulting from dissociation of a bigger molecule. And we're going to learn about association and dissociation of these proteins. They don't exist as monomers. They associate in a quaternary structure form. But depending on the pH and the ionic strength and the temperature, they might dissociate from their bigger quaternary structure to smaller structures. In their associated state, Vicilin is 7s. In their, again, Legumin in their associated state, they're 11s. If they grow from there, they could, due to heat, for example, or any induced disulfide linkage, for example, you can form a 15s polymer. It's an aggregate of the smaller components. So the 11s to the 7s ratio varies between species. So for example, in soybeans, different species, this ratio of legumin to Vicilin can range from 0.5 to 1.7, and in regular common bean, it can range from 0.3 to 0.5, the ratio of 11s to 7s. So varying the ratio will impact the final application. So we will talk about Vicilin structure, and when we talk about the structure, we'll find out that Vicilin is a better emulsifier, or have induced better emulsification property, whereas the 11s has a better gelation property. So if we look at soy protein specifically, it is added in food for its emulsification and gelation property. It's not really added for foaming, or added for viscoelasticity, or added for solubility. It's really added for this kind of functionality. And depending on the variety, some variety might have better emulsification, others might have better gelation, depending on the ratio of these two proteins. So, looking further into characterizing these proteins, they are oligomers. That means they're not monomers. They are an associate that form multiple monomers associate and form an oligomer. High number, okay, there's a high percentage of hydrophobic amino acid. Sometimes the percentage of hydrophobicity in certain subunits can be greater than 40% of the total globulin in soybeans. They are high in acidic amino acids, especially aspartic acid, and we'll talk about the role of that and how it impacts solubility, for example, at different pH and ionic strength. High homology in amino acid sequence, meaning the same sequences repeat in different subunits. So same amino acid sequence. So the homology means you have similar amino acid sequences that repeat in the different subunits. And then the molecular structure and ratio we talked about of 11s to 7s impact functionality, mainly emulsification and gelation. Okay, so what do you want to know here? You want to know, of course, legumin is your 11s. So there is a range, but the average is 11s in sedimentation coefficient. The molecular weight ranges from 300 to 400, but that's not of a one monomer or one molecular protein. That's of the associated oligomer. It can be that high. High in acidic amino acid and arginine. I'll show you composition in a minute. They are low in sulfur containing, but they do contain sulfur. They have on average around 4 cysteine in one monomer. And sometimes they're glycosylated, but not really. Legumin is not as glycosylated as Vicilin, for example. It has much less than 1% saccharide. Lupin, for example, is a legumin, that is glycosylated, but very small percentage of that.
I have a question.
Yeah.
Did you just say lupin was a type of legume, a type of protein?
Yeah.
Is there such thing as like a lupin
Bean.
seed?
Yeah, there is a lupin bean. Yeah. Yeah. Okay, so looking at, here's where it gets a little complex in structure, and I'll try to simplify it as much as I can. So the 11S molecule of legumin is composed of 6 monomers, but the monomer is made up of 2 subunits. Confused? You should be. Okay, so look at this black and light blue. These are linked by this red bond. This is a disulfide linkage. So the black and the blue are linked together by disulfide linkage. This is one subunit and this is another. Together they are a monomer. Okay, so 2 subunits form a monomer. Alright, the black one is acidic subunit, the light blue one is basic. So this is just an animation, I'd like to visualize it. So you have the larger circle is the 35 kilodalton and the smaller one is your 25 kilodalton. So you have 2 subunits, an acidic which has a molecular weight that is larger than the basic. Together, they're linked via disulfide linkage, 1 or 2, depending on the variants, the genetic variant. And because they're linked together via disulfide bond, they're called a monomer.
Is that the case in a lot of other things where if it's linked that way?
What's that?
Is that the case for others where if it's linked with cysteine bonds,
It's a monomer.
it's a monomer?
Yeah, in this case, yes. Just think about legumin.
Okay.
Legumin as part of the legume protein. Yeah. There is like I said, there is a high homology of the amino acid sequences. It's really high. 34 to 67% of the sequences are identical. All right. So here we go. Monomers are arranged in trimer. So this is 1, 2, 3 monomers, right? So 1, 2, 3. And then this black line here represent hydrophobic interactions. So the red represent disulfide, the black here represent hydrophobic interactions. So I'm trying to help you visualize this by giving different colors to the subunits and different colors to the bond. Okay? So you have a trimer. Each trimer is a hexagon. So it's a hexagon because it has 6 subunits. Okay? 2 identical hexagons are arranged one on top of the other via electrostatic and hydrogen bonding. So here we go. So your 11S is as such. 11S at, let's say, pH 7 and ionic strength of 0.5, it's associated like that. 2 trimers are associated by electrostatic. These dotted lines here now represent electrostatic and hydrogen bonding. So the trimer can also be called a hexagon, right? And the 2 trimers are called hexamer. Just in case you see these terminology. Okay, so this is a hexamer. One of them is a hexagon, and in other words is a trimer. Trimer because you have 3 monomers. A monomer, 2 subunits. Very simple. You know this, right?
Yeah, I was like, you might see this next week. FYI. It's coming back.
Coming back to you. Okay. So, I think you've seen this before when we talked about, running, reducing and non-reducing gel. If you run this legumin as is on a gel like that, because of the fact that there are disulfide linkages, it's going to smear up because these subunits are not going to be separated. When you reduce it, you will clearly see this is your basic and this is your acidic. Sorry, this is acidic, this is basic subunit. The acidic is of a larger molecular weight than the basic. But you have to really reduce the disulfide linkages in order for them to travel as single subunits down your gel. So this is when you reduce, you will see that the Veselen also is made up of 3 different subunits, and the glycinin is made up, or the legumin is made up of 2 major subunits, but there are different variants of the acidic subunits. So you have some secondary structure, 6% alpha helix, a lot of beta sheet, and a lot of random coil structure, and they are globular proteins. So the monomers and those subunits are really globular proteins. So like I said, there are different variants of them. So that you have about 5 different acidic subunits, different varies from one variety to another, or in one variety you might have multiple of them. That's why you see them in one gel. So you see multiple, you see the A3, A1, 2, and 4 chains. But in one variety you might have more of the A1 than the A3, for instance. And then also the basic subunits, there are different variants of it, 1, 2, 3, and 4. They differ in a little bit of the amino acid sequence, and because of that they vary slightly in the molecular weight distribution. And they're grouped in 2 different groups, and don't worry about that too much, the different grouping, but just FYI, the homology within a group is over than 84%. That means the similarity in the amino acid sequence within one group is really high. Yeah, I wouldn't, that I said I wouldn't worry too much about such a detail. It's just that you know how they differ. Okay. Questions? I should change the battery for this. It's not as intense, huh?
Yeah, that, I noticed that.
You noticed it? Okay. I don't know if I have some. So Veselen is the smaller of the 2. It has an average. There is a range of sedimentation coefficient, but the average is 7s. The molecular weight is smaller, obviously, 180 kilodalton. It's mostly glycosylated. That means every possible site that can be glycosylated is glycosylated with saccharides or hexoamine. Hexoamines, that means this is hexo, they have 6 carbons and then an amine group. No sulfur, sometimes very little or no sulfur containing amino acids, so they don't form disulfide linkages. And then they are rich in acidic amino acid but low in arginine. So they're more acidic than legumin, basically. So the Veselen are made up of 3 subunits. They associate together via hydrophobic interactions. They have very similar molecular weight, average is 60 kilodalton, and they have high homology again and some secondary structure, a lot of random coil, and again it's globular protein. So again here, if you want to run them on the gel, here are your Veselen subunits. You have alpha prime, alpha and beta. 3 different subunits looking at beta-conglycinin. It's not showing anymore. You have the molecular weights are very close. It ranges from 72 kilodaltons, 68, and 52. So in soybeans, you have the beta-conglycinin is the main one, but you can also have gamma-conglycinin and basic 7S globulin, but they're not as prominent as your beta-conglycinin in terms of concentration. So these 3 subunits together will give you the average of 180 kilodalton per 1 vicillin. So all 3 subunits, they are low in arginine, right? So they're all are acidic subunits. And this is important for understanding the structure and the functionality later on. You have the alpha prime and alpha are present on the exterior of the association, whereas the beta is in the core. So if we want to look at visually, so let's say this is alpha prime and alpha, and the green is the beta, is mostly in the interior, and the other 2 are taking most of the outside of the associated structure. And like I said, all potential sites are glycosylated. So again, there are several species that have been isolated. So the species mean they differ in the trimer combination. So the trimers can be all made up of beta units, all of alpha, or you can have a combination, alpha prime and 2 betas, or 1 of each type. So it depends again on the variety of the variety and the genetic differences. Okay. So moving on to the impact of structure, the association and dissociation, and the impact on different functionalities as affected by pH, ionic strength, heat, and susceptibility to enzymatic hydrolysis. So like I said, they are associated, right? So they can dissociate based on the environmental conditions. So here I'm just reminding you what the ionic strength is. It's different from the total salt concentration, because in the ionic strength you take into account the concentration of each ion you have and the charge on each ion and the summation of all of the other ions, or all the ions you have in the system. This is just a reminder what is meant by ionic strength. So the association dissociation of the quaternary structure of these proteins impact by ionic strength, pH, temperature, the concentration of protein, presence of denaturing solvent, and whether or not you have a reducing agent. So let's start from here. If you have a really high or low pH and you have a reducing agent, then you're most likely breaking it down to low molecular weight subunits. That's this way. So if you go back this way, going not too high of a pH, not too low of a pH, at a specific ionic strength, you form your 7S. More of that, you form 11S. And if you add more heat and the pH is close to the isoelectric point and the ionic strength is really high, then you form aggregates. And then protein concentration, the higher the protein concentration, the more likely you're going to form larger associates. By inverse conditions here is when you go backward, you have the inverse conditions of no reducing agent, for example, and not so high pH or low pH. So if we want to visualize it, okay, so in the past, some of my figures were produced by students and I gave them extra credits. So if anyone wants to regenerate this animation for extra credits, you're welcome to do so.
How much extra credit?
Everyone does it.
$5, $10. 2 points, 5 points. I would say 5 points. Yeah?
Challenge accepted.
But first come, first serve. Maybe you want
race
you
is
want
on.
to
Race
decide.
is on.
You need to decide who wants to do this. Okay. So here's the structure that I drew for you, the hexamers. So if you increase or decrease the pH, that means you're increasing charge and you're changing the ionic strength. You might, and inducing some temperature change, you might break hydrogen bonding and hydrogen and electrostatic interactions. So you would dissociate the hexamer into a trimer. Okay? So at extreme pH, very high charge and very low concentration or a low concentration of denaturing, so you are reducing it to a monomer. As a reducing agent, you're breaking the monomer into single subunits. Okay? Add heat to that, then you are really opening up the protein. It becomes denatured, not just not associated, denatured. At this point, without a reducing agent, but let's say you have a solvent, a denaturing solvent, or heat, you can denature it, but yet you still have the disulfide linkages here. So you can reverse conditions. So you can oxidize, it goes back here, change the pH, it associate, change the pH ionic strength, it will reassociate into a hexamer. So basically here it illustrates that changing pH, salt or ionic strength, temperature, reducing agent can change the association and can cause denaturation as well, depending on whether you have a denaturing solvent or heat. Okay. So this is common condition for a hexamer. When you have pH 7.6 and ionic strength of 0.5, you have a hexamer. Just not pointing. There. Alright, so when the pH is 7.6 and the ionic strength is 0.01, oh sorry, this is the hexamer, this is the trimer. So at 0.5 you have the hexamer. When you reduce the ion strength, you break some of these bonds and then you will end up having a trimer. The beta-conglycerol condition is again pH 7 and greater than 0.5 ionic strength, you have a trimer. When the ionic strength is lower over a range of pH, you end up getting a higher associate of a 10S, which is a hexamer. I'm not going to expect you to know this details, FYI, to see the impact of ionic strength and pH, but I would like you to know at least the normal structure is pH 7.6 and 0.05, you have the hexamer, and then pH 7 and then the ionic strength greater than 0.5 is your regular trimer. So what impacts the association? If you add urea and STS page, you are breaking hydrophobic interactions, and you are breaking electrostatic and hydrogen bonding. So you will end up having monomers. If you have high acid concentration, you have like charges, you are also causing dissociation into the monomers, and the monomers might denature as well. At really high pH or low pH, your actual globular proteins will denature, but not necessarily disulfide bonds will not break. And then this you might want to update it if you have it on your slides. So glycerin has an average denaturation temperature of around 90 degrees. Beta-conglycine is around 75 degrees. So the glycerin has higher denaturation temperature given the fact that it has disulfide linkages.
So why are there different values in our slides? Is it just, it was previously thought they were both?
Yeah, it's based on previous reports, but the newer reports, yeah, I try to update my slides, so probably I update it after I put up the slides. Okay, so this is actually ran in our labs. We looked at soy protein isolate prepared in our lab. You can see this is a DSC looking at endothermic peaks. If you see endothermic peaks, that means there is an uptake of heat when you place your sample in the DSC and you increase the heat flow over time. Then you will see that if there is an endothermic peak, that means the protein was not denatured, it took up heat and it denatured. If the protein was already denatured, you won't see any endothermic peak. That means it's already been denatured, so it won't take, there won't be a structural change upon applying heat. So we identified that this peak is glycinin by running a purified glycinin fraction, and we identified that this peak is beta-conglycinin when we isolated beta-conglycinin from this soy protein isolate. So the solubility is very typical for a globular protein. So when you have an isoelectric, around the isoelectric point, you'll have the lower solubility. As you increase the pH or decrease the pH, you have higher solubility. And here you have more solubility alkaline condition than acidic conditions due to,
More ionized organisms.
which are
Cysteine air.
Serine.
tyrosine, the one with the weight.
Oh, well, wait, what is it?
Tyrosine.
What about serine?
No,
Serine?
I don't know that structure.
No.
it's ATH code.
No. Oh, well, oh yeah, that's right. The serine, it has an OH group. Yeah, and serine and threonine do. But tyrosine is the one that gets ionized. It's the phenolic ring with the OH get ionized at higher pH. Okay. This is taken from one of the reading material I put up. And this is a nice illustration of the impact of ionic strength and pH and the change in the exposure of the structure on the surface of the protein. And how does that impact solubility? It's interesting. It's a little bit complex. I'll try to simplify as much as I can. So in this paper, they looked at the solubility of glycinin so it's a glycinin fraction, purified glycinin fraction, and they looked at the solubility at ionic strength 0.5, so this would be those squares here. And they looked at the solubility as 0.2, which is those black diamond shapes. And then they looked at the solubility at 0.03, which is the X. So you have really low ionic strength and high ionic strength. So what they noticed based on this curve, they made, they deduced changes in structure. So if we look at 0.5 ionic strength, they say the exterior of this quaternary structure is dominated by the acidic subunits. So the acidic subunits, meaning you have more negatively charged carboxyl groups, right, than the basic groups. And the basic groups are more hydrophobic than the acidic groups. So we have two different things between basic groups and acidic groups. Acidic groups are more negatively charged, and the basic groups are more hydrophobic. Okay? So if we have high ionic strength, the acidic groups are 12 times more exposed than the basic group on the surface. So you have a lot of negative charge and polar groups on the surface. Okay? So what happens here is that at low pH, most of your carboxyl groups are ionized, right? So not ionized, protonated. So you lose your COO minus becomes COOH. So you lose your net charge. That's why you have low solubility. Okay? As you increase the pH, they become deprotonated, and the solubility continues to increase because the acidic groups are dominating the surface, so they dominate the solubility, the impact on solubility. Okay, at lower ionic strength, what happens here now, your basic groups are more exposed. You still have more acidic groups, 4 times more than basic groups at the surface, but you still have more exposure of basic groups on the surface. That means now you have more hydrophobicity a little bit on the surface, and you have less negative charge. So at low pH, you will have the basic groups ionized, having an H3+, so they are ionized, so you have a positive charge. And you have less ions, so they are interacting, the water interacts more with the protein. So you have more positive charge here, and you have more interaction with water, so you have good solubility. Okay, now reduce the pH to the isoelectric point. So now you are losing charge. So you lose solubility. And then when the pH goes up again, then it becomes negatively charged, and then more interaction with water. So it's a typical solubility curve. Again, this is highly impacted by what is exposed on the surface of the protein. And what makes the protein exposed differently is the amount of salt. So I will explain this figure and I will stop after that. So bear with me a little bit. So here is the glycinin. They looked at not just glycinin fraction, they looked at isolated acidic subunits and isolated basic subunits. So this is the acidic subunit, this is the basic subunit, and they looked at glycinin that is not being fractionated. That means it has both acidic and basic subunits. So what's the striking difference that you see?
Almost done.
Striking difference that you see is that the basic subunits behave very differently from when it is with than when it is glycinin, that is a combined fraction. But the acidic subunit behaves very similar to the glycinin. Why? Based on what we said in the previous.
Acidic is more soluble than the hydro.
They're more exposed to the surface. So the solubility is more influenced on what is on the surface. So the acidic subunits in glycine are more on the surface. So that's why the solubility resembles the solubility of glycine. Okay? Now look at the basic subunits. We said it's more hydrophobic, right? Let's look at the amino acid distribution. So the amino acid distribution in basic subunits, look at the blue ones. The blue, the alanine, valine, leucine, and phenylalanine is much higher in the basic subunit. So you have very high hydrophobic groups compared to the acidic subunits. So in the basic subunit, you have about 28% of the amino acids are hydrophobic. In acidic, 20% of the amino acids are hydrophobic. When you have 28%, this is above the critical percentage for hydrophobicity. Hydrophobicity will dominate. Even if you have charge, hydrophobicity will dominate. It's really high, above a critical percent. Whereas 20% is below the critical percent of hydrophobicity. Give us 5 minutes, please, or 2 minutes. Gosh, you guys are anxious. Give me 5 minutes. I'm stressed out. I'm going to stop and redo next time. It's complex. I really want you to understand this. I'm not going to rush it. All right, never mind. I'll continue next time. Wow, they're very anxious to get in class.